Hey everybody, I hope you guys are healthy. This is our week 2 or part 2 PowerPoint presentation for genetics. We are starting monohybrid crosses. So we're doing Punnett squares now. There's going to be a lot of video clips in this PowerPoint as well as video tutorials posted. So please make sure you guys are reviewing those. So a Punnett square, it's a chart that shows all possible allele combinations for offspring. So basically you're going to be given the genotypes of two parents, and then the inside the Punnett square is going to show you what the possibility of the genotype of the children are. So by using the genotype, you should then be able to find the phenotype. So down here at the bottom, this is a monohybrid cross, which is a cross between two parents, but it's only involving one trait. So that's where the mono comes from, one trait. So in this case, this could be eye color. When we get to pea plants, it could be plant height. It's only one trait. So when you're making a Punnett square with one trait, it's a 2 column by 2 row table. So it's going to have 4 boxes within it. So each square inside here represents the possible offspring. You always put the parents' genotypes here at the top. And because of the law of segregation, you only can put one allele over each box. Same thing here, one allele over or next to each box. So you can put the father up top here. You could put the mother up top here. It doesn't matter the position where you put the mother or father, okay? So what happens is when you bring down the gamete from the father and you bring over the gamete from the mother, this then would be the genotype of the child. Notice how they're always putting capitals before lowercases to show the law of dominance. Here is an example of a monohybrid cross involving earlobes. So they're telling you at the top, you have to read the information that's given. So it says free earlobes, capital E, that means it's dominant, to attached earlobes, which is lowercase e, which means it's recessive. You're going to cross between two people who have a genotype that is heterozygous for free earlobes. So you guys have to remember from last week's PowerPoint, capital E, lowercase e represents heterozygous. Heterozygous means different. So they're going to have one capital E, one lowercase e, and then you're going to cross it with another parent that is capital E, lowercase e. So they're both heterozygous. So then what you're going to do is you're going to place the genotypes of each parent onto the Punnett square. So here's dad's and here's mom. Remember, it doesn't matter if you put mom up top or dad's on the side. Okay? Then what you do is you bring the gametes or the alleles together. Again, you put capital before lowercase to show dominance. So this would be the possible genotypes of the children or the offspring. So then what ends up happening is you have to go back to see what your letters are representing to then figure out the phenotypes. Okay, so it says what percent of the offspring will have free earlobes. So if you go up top here, free earlobes. So from last week's PowerPoint, it's dominant, which means it can be homozygous dominant, so that's 2 capital E's, or heterozygous, 1 capital E, 1 lowercase e. Remember, if you just have 1 capital E, it's showing dominance, which means that child would have free earlobes. So if we look here in the Punnett square, here's 1, here's 2, here's 3. So then it would be 3 out of 4, which then is 75%. So the next one says, what percent will have attached earlobes? So again, you have to use the information that's given. They're telling you attached earlobes is lowercase e. So the only way that you can have recessive is if it's homozygous recessive. So that means 2 lowercase e's. So right here, 2 lowercase e's. So that would be 1 out of 4, which then is 25%. So when you're writing your genotypic ratios, a lot of the problems are going to ask you to write genotypic ratios. Okay, the genotypic ratio describes the number of times a genotype would appear in the offspring. So how many capital E's are there? How many capital E lowercases are there? How many lowercase e's are there? So in this case, you start at the top left box and we would say, okay, there's 1 capital E. So that's why it's written here, 1 of those. Then you go to the next boxes. Oh, there's 2 capital E lowercases and then 1 lowercase e. Okay, so you always do, when you're writing your genotypic ratio, for a monohybrid cross, you always put the homozygous dominant first, then heterozygous form, and then homozygous recessive form. Now, if after you write the genotypic ratio, then use that to write the phenotypic ratio. So remember, phenotype is the appearance or the actual physical trait. So a phenotypic ratio represents a relationship between the different physical characteristics and how often they occur. So in this case, it would be how many free earlobes, since free earlobes is dominant. So you would write the dominant one first, to then how many attached earlobes, which is recessive. So in this case, the phenotypic ratio would be 3 free earlobes. So remember, that's this one, capital E, and then the 2 heterozygous forms. These all represent somebody that would have free earlobes. And then the only one here would be the attached free earlobes. So it would be 3 free earlobes to then the ratio sign to 1 attached earlobes. Here's another example, and it goes through the different steps of how to actually solve a monohybrid Punnett square cross. So we're talking about dimples versus no dimples. So it's one trait still. That's why you have 4-box Punnett square. So it's telling you that capital D stands for dimples, so that means it's dominant, and then lowercase d is recessive, so no dimples. So it says cross a man that's heterozygous for dimples. So if we're looking at heterozygous, you guys remember from last week's PowerPoint, that means you have 1 capital, 1 lowercase. So there's dad's genotype. Then it's crossed with a female who has no dimples. Well, the only way to be recessive is having 2 lowercases. So here's mom's no dimples, okay? You then put the 2 alleles together, and then remember, you're going to put dominant before recessive. So the first step is to find the genotypes of the parents and place them on the Punnett square. So that's what we did here. Now, once you put the parents on, now we're going to do step 2. Fill in the genotypes of the possible offspring. So that would be filling out here. Now, remember, you put capital before lowercase to represent dominance. So that's step 2. Okay, step 3 then would be interpret the results of the Punnett square by answering the questions being asked. So that would mean genotypic ratio, phenotypic ratio, and then whether or not there is any probability questions. So what is the chance of, or the percentage of? So here's some questions that would be interpreting the Punnett square. So what is the genotypic ratio? So remember, the genotype is the letter combinations. So you would say, in this case, there's no capital D's. So you can you can put 0 capital D's on all of your worksheets. And then heterozygous comes second. So there's 2 of those. And then there's 2 lowercase d's. So the genotypic ratio would look like this. 2 big D, little d's to 2 little d's. Next question would be, what is the phenotypic ratio? So what is the actual appearance? So we know if there's 1 capital D, that means that person has dimples. So right here, we have 2 offspring or possible children with dimples, and then this would be 2 no dimples. Next question. What is the probability or chance of having a child with no dimples? So for probability, remember, you can write it as a fraction or as a percent. So in this case, no dimples. No dimples would be these 2 children. So you would say 2 out of 4 or 50%. So that was an example of a monohybrid crosses that we just did. A dihybrid cross now is a 2-factor cross. So here's the Punnett square. It is a 4x4 Punnett square, so it's 16 possible offspring. So that means when you're doing your probability, it should always be out of 16. Mendel wondered if traits were inherited together. So this is where that law of independent assortment comes into play. Would a tall plant with round seeds pass both traits on together? So the 2 traits would be plant height and then seeds. Are they going to be round or wrinkled? So that's 2 traits. Mendel's experiment showed that the traits are inherited independent of each other, which means you can have a tall plant with round seeds, or you can have a tall plant with wrinkled seeds, or you can have a short plant with round seeds and a short plant with wrinkled seeds. Okay, so they're totally independent of each other. Now, there's a lot of resources that have been posted about solving dihybrid crosses. So you can solve them 2 ways. either the long way, which is the 16 box Punnett square, or you can do it the shortcut way where you actually make 2 monohybrid crosses and then you cross multiply. So make sure you guys are checking out the document that I posted along with all the video clips on how to do those as you're working through. If anybody has any questions, again, please make sure you guys email me.